Thank you, Joy, and go ahead.
Hello, everyone, and thank you for attending today's seminar. As Frances said, my name is Joy Aho, and I'm the Manager of Stem Cell Research Product Development here at Bio-Techne. And today I'm going to talk to you to give you some information about how to optimize and standardize stem cell differentiation. So I just wanted to start out by just introducing the name Bio-Techne in case you weren't familiar with it. And really what this allows us to be able to do is provide a bunch of different product types for you under one umbrella, which is this Bio-Techne umbrella. And it still has the R&D systems and Novice and Tocriscreen and Protein Simple brands that you might be familiar with. But it allows us to group these all together under one umbrella and provide a variety of solutions that kind of synergize together to help enhance your workflow. So directed differentiation of stem cells is really the reason that stem cells are such a powerful tool. The fact that they can differentiate into basically any cell type provides us a valuable tool to be able to look at a variety of different things, including investigating developmental mechanisms, understanding disease mechanisms and drug efficacy and toxicity, and hopefully eventually developing these protocols and cell types to be able to be used in regenerative medicine. Unfortunately, there's a variety of challenges that are involved in this process, which you may or may not be aware of if you've done them, is that they can involve very long and complicated protocols, and there can be a lot of variability across different cell lines. And this can be due to a variety of reasons, some of which can include inconsistent reagent quality as well as inconsistent cell quality. So today what I'm going to focus on are in three main areas. The first is talking about strategies to be able to allow you to more easily develop your own custom protocols, and that includes looking at recombinant proteins, small molecules, and basal media supplements, as well as then looking at some of the differentiation kits that we've been developing to provide consistent, efficient, and complete kits to reduce that barrier to allow you to go into some of these areas and use this as a tool. And then finally, I'll be talking about some tools that you can use for then investigating these cell types, either when you're developing your own protocols or to investigate some of the downstream mechanisms that you might be interested in. So I'm going to start with looking at strategies for standardized differentiation of custom protocols. And the first thing I want to talk about are our ProDots proteins. So this you're likely familiar with R&D systems for their proteins in the stem cell arena, but this is kind of a new thing that we've developed, and it's more geared towards cell culture. So what these proteins are our standard proteins. They're lyophilized into a small ProDot, is what we call them. And as you can see in the video that I show here, they basically dissolve right into your culture media. So it reduces a lot of the time you can spend aliquoting. You have greater confidence that you're getting all the protein out of the vial because you can just roll it right into your media. And it also is designed so that you don't have to worry about potential loss of activity from freeze-thaw because they're all aliquoted out into these nice little dots that you can just roll into your media. So we have a variety of these available, the majority of which are geared towards stem cell researchers. So it can really just kind of help ease your workflow a little bit. So in addition to proteins, small molecules are obviously a very important part to developing your own protocols. And with Tocriscreen Bioscience, we have a variety of these available, many of which are unique to Tocriscreen. And these are important because they can really allow you to target specific signaling pathways and enhance the differentiation efficiency in your protocols. And what I wanted to focus on today are some of the screening libraries available and show you how these really can be used to develop your own protocols. So the Tocriscreen compound libraries are pre-dissolved small molecules at a 10 millimolar concentration, and they're in an easy-to-use 96-well rack format. And they come in a variety of different flavors depending on the signaling pathways that you might be interested in. And what I'm going to focus on today is giving you an example of how we were able to use these libraries to be able to enhance a protocol. So in this example, we did a collaboration with Michael Kaiba's group at the University of Minnesota, where we were looking for molecules that could enhance differentiation of embryonic stem cells either into cardiomyocytes or skeletal muscle. And the way that this was done was using a MESP1 inducible mouse embryonic stem cell line that was developed in the Kaiba lab. And these will push the embryonic, when induced, will push the embryonic stem cells toward a mesodermal lineage. Once these are induced, they are treated with a variety of different compounds from the stem cell toolbox available from Tocriscreen, and then they were screened for their enhancement of differentiation into either cardiomyocytes or skeletal muscle by immunocytochemistry for MHC and myogenin. So here shows you some of that data. You can see in the upper left panel would be the baseline for cardiac differentiation of these MESP1 induced cells. And then they're treated with a variety of different compounds, and you can see the 4 here shown on the left and middle side. You can see great enhancement of the differentiation using these different small molecules. Additionally, we can find similar pattern with skeletal muscle. Again, on the upper left, you can see the baseline differentiation with the skeletal muscle, and then we can greatly enhance this differentiation using small molecules that we can find from the toolbox. We can also go back and then look at these and look at the different signaling pathways that may be involved, and that can help us understand more about the mechanisms involved in these different differentiations during development. So in addition to having the proteins and the small molecules to be able to optimize your protocols, you also, most protocols will require a basal media supplement, and this just to ensure that your cells are going to stay nice and healthy and happy. And 2 of the main basal media supplements that are used in many protocols are N2 and B27. Our version of B27 is, we actually call N21 max. So we've taken these formulations and we've optimized them from the original published formulations, and they're also available in a variety of different flavors. So our N2 Plus and N2 Max are actually basically the exact same thing. The only difference is their source of insulin. And then N21 we have available both without insulin or without vitamin A, and depending on your protocol of interest, these can greatly enhance, changing some of these different flavors can greatly enhance your protocols. So just to show you an example of that, here's differentiation of human embryonic stem cells into PDX1 positive pancreatic progenitors, and this was done using either a competitor product or an N21 Max product. And you can see here that we can see enhancement by flow cytometry, looking at expression of PDX1 in these cells, that, and then we can quantitate that, as you can see in the graph on the right-hand side. So now, in addition to having the reagents available for you to customize your own protocols, we've also been developing a variety of differentiation kits. And the great thing that we have at our disposal here at Bio-Techne is that we do have a ton of recombinant proteins. Obviously, we have the biggest selection available on the market, as well as the Tocriscreen Bioscience and antibody collection available to us. So we are able to take all of these reagents and screen them and put them together in optimized kits to allow you to easily differentiate your cells into different lineages. And we do have these available for ectoderm, mesoderm, endoderm, and cardiomyocytes. And newly released are for hepatocytes. So this graphic here gives you kind of an idea of what the majority of our differentiation kits are, how they're structured. And they're all monolayer based. And basically they're going to come with basal media supplements and cocktails that you mix and match for different stages to be able to guide your stem cells through the proper developmental stages to reach the terminally differentiated cells characteristic of that kit. And the timeline will depend on what cell type that you're interested in. You can see in the bottom left-hand corner that gives you an idea. In addition to these, each kit also comes with an antibody so that you can confirm your differentiation and look at efficiency. And this can help you when you're looking from cell line to cell line, as you might see some differences depending on the potency of your different cell lines. So I just wanted to highlight some data around a couple of these different kits. So here shows you is the Cardiomyocyte differentiation kit, and it comes with the components that are listed on the left-hand side, and you can see that we can detect nice beating, or we're able to see nice beating sheets of cells. In addition to the cocktails, it comes with a cardiac troponin T antibody for you to look at expression of cardiac markers in these cells. So in addition to just being able to visually see the beating, we also verify differentiation looking at different cardiac markers, and you can see here in these cells we can see double expression for cardiac troponin T and I. We can also look at this by flow cytometry to be able to better quantitate our differentiation efficiency, and we can generally see upwards of 70% differentiation when we're using the kit. Another concern with differentiation protocols is that you can see differences when you're starting with different media formulations for your initial culture. So what we've also had is other groups test out the kit, and in this example, a group at the Mayo Clinic used the kit to differentiate some of their IPS lines, and these lines were grown in a common media M teaser and found that they were also able to differentiate very robustly. And you can see here in this image, these are, again, double positive stain for troponin T and troponin I. So finally, in addition to being able to differentiate these cells, you can also then maintain them over time. So we've developed a media supplement to allow you to keep them in culture over time and continue to do whatever analysis you have of interest. So these are cells that have been kept in culture up to 80 days. We've also gone as long as 200 days in culture. We can also replate these cells once they've been differentiated. And you can see that in the staining on the right-hand side, that we're still able to detect nice expression of cardiac-specific markers. So now moving on to our newest kit, which is our hepatocyte differentiation kit. Again, shown on the left-hand side, demonstrates the different media supplements and cocktails that are included, as well as this kit comes with a serum albumin antibody to be able to detect differentiation. We can also see nice hepatocyte-specific characteristic morphology, as you can see in the bright field image on the right-hand side of your screen. In addition to that morphology, we've also looked at a variety of different hepatocyte-specific markers, as you can see in the immunocytochemistry here, and we can see robust expression of those in the differentiated cells. Really, ultimately, what's extremely important with hepatocytes is making sure that they're able to be functional, and here are a couple different assays where we're able to assess that. We're able to see albumin secretion as detected by ELISA, comparing undifferentiated embryonic stem cells to differentiated hepatocytes, as well as urea secretion, as you can see on the right-hand side, of both embryonic stem cell-derived and iPS-derived hepatocytes. An additional functional verification is making sure that these cells are able to express functional P450 enzymes. And in this assay, we were able to detect activity of CYP1A1 by treating cells with beta-netfloflavone to induce that activity, and then looking at metabolism of ethoxyresorphin into the fluorescent byproduct. And we can detect nice metabolism of that over time. In addition to CYP1A1, we've also looked at CYP3A4 using a luciferase-based assay, and we can see nice induction of that with dexamethasone and rifampicin. So now that you have the tools to be able to differentiate your cells into these different cell types, it's really important to then have tools to be able to investigate them easily, because you really want to get at the answers to whatever questions you might be interested in your research. So some of the important questions that you might have for investigating these cells are, first of all, characterization of the cells. So it's really important to know you have the right cell type. Are they expressing the correct markers? Are they functional, like some of the data that I showed you for the differentiation kits? But additionally, these type of tools can also allow you and help you to optimize your own custom protocols. Ultimately, it's important to look at some of the important questions are looking at disease modeling, so what might be differences between disease patient cells versus healthy controls, as well as drug screening and discovery, and using these cells and their derivative cell types and seeing how drugs may alter the phenotype of the cells. So to show you some examples of that, here we can look at modeling development in vitro, and in this example we've differentiated pluripotent stem cells into pancreatic progenitors, and by using antibodies for immunocytochemistry we can follow development and look at how the cells are differentiating into definitive endoderm and then into foregut and then pancreatic progenitors, and this allows us to maybe look at some of the signaling pathways that might be going on and model development in vitro. There's a variety of ways that we can also look at drug screening. Initially, just looking at cell viability can be very important. In this example, we've looked at differentiated cardiomyocytes that have been exposed to cardiotoxic small molecules, staurosporine and doxazosin, and we're able to detect just using a redox dye, rizazirin, and see, and we can see the viability loss after treatment with these small molecules. Additionally, using as an additional example, we can look at cell viability in hepatocytes using a similar redox dye assay, and we can treat the cells with different doses of acetaminophen, and we're able to detect a dose-responsive decrease in viability after exposure to this drug. In addition to just looking at viability, there's a variety of other factors that can be used to look at and assess possible disease mechanisms. One of which, and shown here, is looking at calcium imaging in cardiomyocytes, and you can be able to assess contraction rates. And in this example, we've taken pluripotent stem cell differentiated cardiomyocytes, and you can see the beat rate pattern in the untreated cells, and then slowing down once you treat them with a beta-adrenergic antagonist, propranolol, and then we can speed that beat rate back up with isoproterenol. And once you do this, you can put these into imaging programs, and you're able to quantitate those beat rates, as you can see here in the graph, and in the contraction rates in the table on the bottom. Just as another example, we can also detect dose responses in these cells. So these are, again, cardiomyocytes that have been treated with verapamil, which is a calcium channel inhibitor, and we can see the slowdown at 10 micromolar, and then you'll see once we hit this 30 micromolar, we're basically stopping all beating of the cells. So in addition to looking at calcium imaging, another option is to be able to look at some of the biomarkers that are characteristic of your disease of interest. And here in this example, we're using ELISAs to be able to look at a marker for hypertrophy. So these are cardiomyocytes that have been treated with endothelin-1, which is a known to induce cardiac hypertrophy. And then one of the biomarkers is a pro-ANP marker, and we're able to detect a dose-responsive increase in that with increasing doses of endothelin-1. So in addition to the standard ELISA, I wanted to introduce you, in case you weren't aware of it, to the Luminex technology, which I think is a really powerful tool that a lot of the stem cell community is not aware of. And really how it works is that it's very similar to doing it basically is a sandwich-based ELISA, but it's in a bead-based format that allows you to look at multiple analytes in a single very small sample. So the way it works is you have, as you can see in the image shown on the left-hand side, you have a variety of beads that are coated with different dyes, and each one of those colors is going to be associated with a specific analyte that's then coated with a capture antibody. Then you can use a detection antibody that's labeled with PE to be able to detect the amount of signal in your sample. So you can look at, depending on the format, up to 100 different analytes in a single sample, which I think is an amazing tool that's really underutilized in the stem cell community. And just to give you some sample data using Luminex, in this example we're looking at, again, hypertrophy in endothelin-1 induced cells, and we can see nice induction of endothelin-1 after treatment, or we can see induction of pro-BNP, after treatment with the endothelin-1, and we're not seeing that in the other two drugs that we're treating with. Additionally, we can look at secretion of troponin T, which is a biomarker for heart failure, and we can see that increased dose response we're able to detect using Luminex treating with doxorubicin. So in this slide really is the kind of highlights the power of Luminex. So here looking at different serum samples either from heart failure or from healthy patients, and you can get a nice profile looking at a variety of different analytes to be able to look at what's going on in these cells. And this can be a really nice tool to be able to investigate some downstream mechanisms and find some interesting biomarkers. So obviously using Luminex is a great screening tool, but if you want to do more in-depth analyte analysis, we are the gold standard for ELISAs. And in this example, you can see how the ELISAs can also be a great tool for you to optimize your differentiation protocols. So here we're looking at insulin secretion in pluripotent stem cell-derived pancreatic beta cells, and we've differentiated 2 different iPS lines as well as a human embryonic stem cell line using 2 different differentiation protocol strategies, condition 1 and condition 2, and we're able to look at how the levels of insulin secretion are changing depending on the protocol that we're using. So hopefully what I've been able to tell you about today is some of the workflow solutions that we've been developing here at Bio-Techne. Because we have all of these great tools at our fingertips in the stem cell group, we're able to really build some great kits to allow it to enhance your workflow. And today I really focused on the differentiation and investigation things that we've been developing. But I also just wanted to highlight that we do have a variety of products and medias available for both for isolation and culture, as well as to really easily verify your stem cells before going into differentiation. And we've been working really hard to develop easy-to-use kits to allow you to verify your stem cells to hopefully increase your reproducibility in your differentiation experiments, since they are really labor and time and reagent intensive. So I really encourage you to visit our website and check out all of the things that we have been developing for stem cells. And I would be happy to take any questions. And thank you again.
Thank you very much, Joy. I'm going to anticipate the most frequently asked question that we have from the audience and say, yes, we have recorded the webinar so that everyone will have the opportunity to be able to watch and listen again later, or indeed to be able to pass on the hyperlink to other people who you think may be interested in listening to what Joy's had to say at this webinar. If you have any questions, then please feel free to type them into the question bar below. We'll watch and see what pops up. No? Joy, I'm just going to read a couple of questions out to you that perhaps people would be interested in hearing your answer to. So George has asked what type of equipment we need for using the Luminex assays, and we also have another question about information about the multiplex assay system. So there's kind of 2 related questions there about the Luminex assays and the equipment needed.
Sure. It does require a Luminex type reader, and there's different vendors available for those. But I would say, since the majority of people may not be aware of the technology, they may not realize that there probably is one down the hall from them. So especially if you are in an academic lab, the majority of academic labs that I've been talking to, it turns out that there is one, in maybe a core facility down the hall. A lot of institutions will have at least one that can be shared, or it might be in a different department, but would be, really easy for you to go over and use.
Okay, thanks for that. So we had a question regarding pancreatic cell differentiation, and are we working on a kit to enable customers to be able to differentiate pancreatic cells?
It's a possibility. At this time, we're not sure.
Okay. Similar question about differentiating cells into neural stem cells from induced pluripotent stem cells.
Yes, and that is definitely something we are interested in and working on right now in our group. And definitely, if this is, we're always looking for people to test out things that we've developed. So if that is something that you're interested in, you know, definitely let us know.
Okay, so another question going back to Luminex. Can the Luminex system be used to analyze cells in situ, or does this have to require suspending cells? Which I think is probably not quite the application of Luminex. If you could just maybe go over that again, Joy.
Sure, yeah, so it's, you know, it's similar to an ELISA. So really what you would want to be profiling is likely the culture media from your cells, so looking at profiles in the that are being secreted from these cells that you can detect, like I showed, you know, insulin and albumin and things like that are the same type of things that you can detect with Luminex. It is possible to look at cell lysates as well, would be another option, but you wouldn't be looking at an actual like whole or live cell.
Mm-hmm. Okay, I think that clears that up. Well, hopefully. I'll just point out actually at this point that any questions that we don't get to, we will actually be answering by email after this. So don't worry if your question doesn't get answered verbally right now. We will follow up, so nothing is left unanswered. There was another question about the differentiation kits. Can you tell us more about the kits for ectodermal differentiation?
So the kit for ectoderm takes the cells to very early ectoderm, so it's a very short 5-day protocol. And why it was originally developed to be able to do a quick, easy verification of your stem cells. So if you're interested in looking at early ectoderm and making sure your cells are able to go down that lineage. It could also be used if you are interested in that very early ectoderm. So we are able to detect OTX2 expression, as well as starting to get some of the more neural ectoderm markers such as SOX1. But then you could take those cells, and if you are interested, you know, using that kit and then going further down using some more customized protocols.
Okay. Obviously, the differentiation kits are of great interest. Can you obtain brown adipocytes from induced pluripotent stem cells?
So we have not tried that in our hands. I'm trying to think if I've seen anything in the literature. I mean, really, you know, by definition, you should be able to. It's just trying to figure out the strategies to be able to do that. We are always looking for, you know, different interest in what people might be interested in to have a differentiation kit. So that's another thing that I'm always looking for feedback. So if that's something that you think would be really interesting and useful to the field, I would love to get that feedback, and we can look at possible strategies to be able to develop that protocol.
Mm-hmm. Another similar question was about the differentiation of stem cells into retinal cells or inner ear by using various different molecules to stimulate the differentiation.
Yes, and we have played around with that a little bit. We aren't really close to a kit, but we have been able to get some early otic differentiation. But that's another thing that it's just getting enough feedback that would be of interest to the field to be able to put in the resources to start developing a full kit. But we do have, you know, just using our proteins and small molecules, there are some protocols available in the literature that you can use some of the tools that I mentioned in that first part of the talk to be able to start developing those protocols.
Okay. There's a question here about the differentiation protocol. Is there a difference in the protocol that you'd use between an embryonic stem cell and an induced pluripotent stem cell?
No. So the protocols that we use, you may, if there see some differences depending on the cell line, but that could be differences between different embryonic stem cell lines as well as different IPS lines. Depending on how they grow, if they grow really slow, you might have to increase your starting plating population, different things like that. But you should, in general, be able to at least start with a very similar protocol between the two cell lines, cell types.
There was a question here about progenitor cells. I'm not quite sure which lineage progenitor is in question. How do you use kits to generate progenitor cells? So I'm not quite sure if that question is very clear to you, Joy, is it?
Yeah, it could really depend on what progenitor you're talking about.
Yeah.
There's definitely there is a ton of literature out there of protocols that, using the same strategies with the proteins and small molecules and basal media supplements to be able to specifically differentiate into a variety of different progenitors.
Yeah.
And then maintaining them at that stage.
Okay. I think we'll have to follow up that one by email to get to the bottom of that one. Thank you. Okay, we have another question just popped up. Which of your kits require monolayer of single cells or embryoid bodies as the starting material? So does it make a real difference, Joyce, whether you've got a monolayer or an embryoid body?
It can make a difference. All of our kits are designed for monolayer culture. So I think that takes a little bit of the variability out when you can do it in a little bit easier monolayer culture, plating them. Generally, most of our protocols, you want to plate them at a density for that cell line that they will be, 80% confluent the next day, so you can then start the differentiation.
Okay. So just to reassure anybody who feels their question wasn't answered adequately, we will enter into an email conversation with people who've asked questions, so it does give you the opportunity to get more detail if we didn't fully answer the question. And to reiterate, we have recorded this, so this will be available online, and we'll get that hyperlink to you as soon as possible. So after that, thank you very much, Joy. We've taken up more than half an hour, so I guess we should wrap up. Thank you for everybody for attending, and thank you, Joy.
Thank you. Thank you, everyone.
Thank you.